Autophagy, the process by which cells clean out damaged components and recycle them for energy or rebuilding, is one of the body's most powerful natural defenses against aging. It is critical for maintaining cellular homeostasis, preventing the accumulation of dysfunctional proteins, and ensuring the health of mitochondria and other organelles. While much of the focus in autophagy research has centered on nutrient-sensing pathways such as mTOR and AMPK, there is growing appreciation for the intricate role that vitamin-dependent enzymes play in enabling and regulating these self-repair systems. Vitamins B2, riboflavin, B3, niacin, and B6, pyridoxine, are not merely supportive nutrients. They are essential coenzymes for enzymatic activities that directly influence autophagy efficiency, mitochondrial quality control, and resistance to oxidative stress. Vitamin B2 is a precursor to flavin adenine dinucleotide, FAD, and flavin mononucleotide, FMN, both of which serve as critical cofactors in redox reactions throughout the mitochondria. Enzymes that require FAD, such as those involved in fatty acid oxidation and the electron transport chain, are essential for producing cellular energy from lipids, especially during fasting or carbohydrate restriction when autophagy is most active. When FAD-dependent enzymes are functioning optimally, they not only improve mitochondrial respiration but also limit the leakage of electrons that can produce harmful reactive oxygen species, RLS. This antioxidant control is vital during autophagy, where damaged mitochondria and proteins are broken down, a process that can generate bursts of oxidative stress. By enabling efficient energy production and reducing oxidative load, vitamin B2 dependent enzymes help ensure that autophagy proceeds smoothly without collateral damage. Vitamin B3, through its role in synthesizing NAD+, nicotinamide adenine dinucleotide, is equally central. NAD+ is a key signaling molecule in cellular metabolism, particularly during conditions of low energy such as fasting, exercise, or stress. NAD+ dependent enzymes like sirtuins and poly-ADP-ribose polymerases, PARPs, help regulate gene expression, DNA repair, and mitochondrial function, all of which intersect with autophagy regulation. SIRT1, for instance, deacetylates several autophagy-related proteins, including ATG5, ATG7, and LC3, enhancing their activity and promoting the formation of autophagosomes. Sirtuins also suppress inflammation and stimulate mitochondrial biogenesis, enabling a coordinated cellular response that supports both damage clearance and tissue regeneration. Without sufficient NAD+, and by extension, vitamin B3, this sophisticated control system becomes impaired, leading to sluggish autophagy, greater cellular stress, and accelerated aging. Vitamin B6 plays a slightly different but equally critical role. As a coenzyme in over 100 enzymatic reactions, particularly those involved in amino acid metabolism, B6 helps regulate the availability of key building blocks for cellular repair during and after autophagy. One of the lesser-known aspects of autophagy is that, beyond degrading damaged materials, it supplies raw substrates that cells can repurpose for survival under stress. B6 dependent enzymes help metabolize these substrates, including glutamate, serine, and cysteine, into compounds needed for energy, detoxification, and antioxidant synthesis. For instance, B6 is essential for the synthesis of glutathione, the master antioxidant that protects cells from oxidative stress during autophagic breakdown. Additionally, vitamin B6 influences lysosomal function, ensuring that autophagosomes are properly fused and degraded, an essential step in completing the autophagic cycle. Deficiencies in B6 have been linked to impaired autophagic flux, inflammation, and susceptibility to neurodegenerative disease. The cumulative effect of these vitamin-dependent enzymes is to enhance the efficiency, precision, and safety of autophagy. While nutrient stress may trigger autophagy, it is the enzymatic machinery, powered by these vitamins, that determines how well the process is executed. Without sufficient cofactors, the body may activate autophagy but fail to complete it properly, leading to the accumulation of intermediate waste, energy deficits, or unintended tissue damage. This becomes particularly relevant with age, as nutrient absorption often declines, and the demand for cellular cleanup rises. In aging tissues, ensuring a steady and bioavailable supply of these vitamins may mean the difference between successful cellular recycling and dysfunctional degradation. What makes this strategy compelling from an anti-aging perspective is its ability to work in harmony with other longevity-promoting interventions. Fasting, low-carb diets, and exercise all create favorable conditions for autophagy, but they also place additional stress on the system. If the body is not equipped with the necessary enzymatic cofactors, these interventions can underperform or even backfire. Supplementing or prioritizing foods rich in B2, such as eggs, liver, and almonds, B3, found in poultry, fish, and legumes, and B6 in bananas, chickpeas, and sunflower seeds, can amplify the benefits of these metabolic stressors by ensuring the cellular repair machinery has the resources it needs. In conclusion, the anti-aging benefits of autophagy depend not just on when and how it's activated, but also on the underlying biochemical support that sustains it. Vitamin-dependent enzymes, particularly those relying on B2, B3, and B6, are crucial for maintaining the energy, redox balance, and metabolic flexibility needed for effective cellular cleanup and repair. By ensuring these micronutrients are present in optimal amounts, either through diet or supplementation, we create the conditions for autophagy to work not only harder, but smarter. In the broader picture of longevity, this underscores a powerful principle. Aging well is not just about deprivation or stress, but about equipping the body with the precise tools it needs to renew itself from within.